All right, so good afternoon to all. It's a pleasure to be here, and I would like to thank the organization committee for inviting me. All right, so here's my disclosures. All right, so quickly, I'm going to be touching upon some platform trial methodology as part of this presentation, also briefly reviewing some of the ongoing platforms of neoadjuvant therapy, with a focus obviously on those that touch upon molecularly defined patient populations, and also spend a good portion of this presentation outlining the Canadian experience as based on the CCTG IND-242 neoadjuvant platform. So these are the CANMET roles that are going to be addressed within this presentation. All right. So the standard of care for most medically operable patients with early stage non-small cell lung cancer is surgery combined with either neoadjuvant or adjuvant chemoimmunotherapy. And obviously following the success of targeted therapy in the metastatic setting, these agents are increasingly being investigated in early stage non-small cell lung cancer. Currently, we only have adjuvant osimertinib and most recently alectinib as approved therapies. But what about neoadjuvant targeted therapies? Well, the neoadjuvant approach in itself certainly has important potential benefits, but most of the trials that are currently ongoing are mostly focusing on either EGFR mutant disease or ALK fusion disease. And as far as published trials of neoadjuvant targeted therapy, as can be seen on this slide, there's actually very few of them. Most have actually studied older generation drugs, and most have focused on either EGFR or ALK alterations. Now, one way that we have to actually become more efficient in bringing drugs from phase 2 trials to larger phase 3 initiatives are master protocols. Now, according to the FDA, master protocols are essentially trials that have, well, multiple arms or sub-studies. There's basically 3 major examples of such master protocols. There's the basket trial, where obviously you're going to study one treatment or drug among different conditions or disease sites. And basically, umbrellas and platforms are the exact opposite, meaning you're going to be studying one condition or disease site and then having multiple sub-studies that are going to be studying different treatments. The subtle difference between umbrella and platform trial is essentially that umbrellas are supposed to be offering all of their sub-studies all at once at the same time. There's many theoretical advantages for platform trials, as depicted on this slide. I would certainly summarize those by saying that it's about mainly shared resources. There's also a factor, logistically speaking, about being able to activate new sub-studies a bit quicker, because these are usually going to be treated as protocol amendments, as opposed to starting a brand new trial. There's also advantages for patients. If your platform trial is actually incorporating a randomization component and a control arm, where basically because there's more sub-studies being involved, patients have actually less of a chance of being assigned to the control arm. Challenges, yeah, there are certainly a few of them. Briefly summarized, it's about complexity. Increased sometimes startup time for the initial setup of the platform, and it needs certainly high coordination between different stakeholders to put these together. So what are the completed or ongoing platform trials that we've seen so far? There aren't actually that many, and most have focused on either IO agents or IO in combination with chemotherapy. Most of you are going to recognize this landmark paper by Dr. Ford and colleagues, which was published in the New England in 2018, but that was actually part of a platform. Nivolumab alone was published first, and then there was an ipilimumab with nivolumab arm published a bit later in JITC, And from what I've been told, there's also a chemoimmunotherapy arm that's part of this trial that has, to my knowledge, not yet been published. Another great example of an academic platform trial would be MD Anderson's NeoStar trial, which results have been published in Nature Medicine successively in 2021 and 2023. The industry also has its own platform trials. One great example is AstraZeneca's NeoCOAST, which was published in Cancer Discovery, looking at durvalumab in combinations with other innovative IO agents. This obviously paved the way for the ongoing NeoCOAST-2 trial, which has some differences, mostly in terms of staging, the fact that it's combining IO agents with chemotherapy and also incorporating adjuvant therapy as well, which makes it more of a peri-operative platform trial. What about platforms that incorporate components for molecularly defined patient populations? There's actually very few of them that we could find. The Lung Cancer Mutation Consortium had initially put out some trials that were studying such oncogenic driver mutations for metastatic disease, but more recently, they have put together this effort for early stage non-small cell lung cancer. This LEADER screening trial is an effort basically trying to see how many patients using this screening approach could basically harbor oncogenic driver mutations. And essentially, this approach for screening is going to be considered feasible if at least 35% of enrolled patients actually do harbor one mutation or the other. This is scheduled to enroll up to 1,000 patients, and it also has TMB as a secondary objective. Now, LEADER is essentially supposed to be a purely screening trial, but it has the Nautica umbrella study, which goes along with it, which basically uses the LEADER screening approach to potentially assign patients to different treatment arms that are depicted on this slide, depending on which biomarkers are being found. Interestingly, results from Nautica 1 have already started being published, at least in abstract form. This comes from last year's World Lung Conference and was showing basically preliminary efficacy and safety data with neoadjuvant alectinib in patients with stage 1B to 3 ALK-positive non-small cell lung cancer. It was shown to be, in 12 evaluable patients, tolerable, and MPR and PCR rates were also quite promising, with 66.7% and 33.3% response rates, respectively. Now, switching gears to our own Canadian experience with the Canadian Cancer Trials Group IND-242 neoadjuvant platform trial in patients with surgically resectable non-small cell lung cancer. So we started working on this about 2 years ago, and there were major features that we wanted to highlight high level for this platform initiative. First of all, this was meant to be mostly for molecularly defined patient populations, basically a signal-finding platform that would justify a larger phase 3 initiatives. So what is shown here is the basic outline of some of these initial sub-studies that we were thinking about. And basically IND-242A, which is the first and sub-study that was activated within the platform, basically showed our initial interest for KRAS mutant disease specifically. So at a high level, we were thinking that basically we could provide targeted therapy neoadjuvantly, followed by surgery, path response assessment, but importantly, the targeted therapy being administered in the neoadjuvant setting wasn't supposed to preclude standard of care chemoimmunotherapy in the adjuvant setting. We had also thought about incorporating an adjuvant portion of targeted therapy for up to 1 year for patients that had at least an MPR, which also represents the primary endpoint, at least as far as this first initial sub-study is concerned. An important part of this platform is also correlative science, as highlighted in the lower boxes of this schema. So lots of planned analysis, both blood and tissue-based. Lastly, I would point out that Indeed, IND242A, the first sub-study, was meant to enroll KRAS G12C positive patients using 2 cycles of Novartis's JDQ443 KRAS G12C inhibitor with a maximum number of enrolled patients of 27. Now, importantly, what has been our experience so far with this platform? What are its strengths? Well, first of all, we have gained a lot of knowledge about the Canadian landscape. What's the uptake of neoadjuvant therapy in general? We have currently 10 major Canadian sites that are activated with this platform, with other sites pending. In terms of reflex molecular testing, well, most sites, I would say, do reflex testing, and so far we have not been asked to provide central testing. The control arm, we currently do not have one, but there's certainly a will and the flexibility to incorporate a control arm in the platform. Patient population remains flexible. As I mentioned, the original intent was mostly to focus on patients with molecularly defined features, but notwithstanding the fact that the platform could still accommodate more wider population, for instance, sub-studies incorporating patients that would be eligible for chemo-IO regimens. As I already mentioned, there's an adjuvant experimental therapy component that's integrated into the platform, so to allow some basic assessment of adjuvant tolerance to targeted therapy, also potentially planning for larger peri-operative phase 3 trials. In terms of accrual, for the first sub-study, admittedly, the KRAS G12C positive population was slower than expected to enroll. However, when John and I sat down to look at our efforts as compared to the other initiatives in neoadjuvant KRAS G12C targeted therapy, we are actually doing better. And as far as translational science is concerned, I've already outlined this is a major component of the platform. We mandate tissue and blood-based collection, so to increase to a maximum the sample size number for these analyses. Now, obviously, throughout the development of this platform, many questions have been raised, and some of them, remain active points of debate and discussions as we move along and plan for further sub-studies. For instance, control arms. What is the ideal control arm for rare molecular patient populations? This is a question mark that remains, and for sure, as was seen throughout this day so far, the platform certainly needs to be adaptive to this rapidly changing treatment landscape. Primary endpoints, whether your preference goes to MPR or PCR, we are potentially expecting lower response rates if only targeted agents are being used. Does this really matter in the end? Do we need to focus on other types of surrogate endpoints that would correlate with overall survival? These are ongoing questions. There is going to be other presentations later on during this conference addressing which type of criteria we should be using for path response. Should it be different when targeted therapies are involved or when IO is actually involved? And obviously, there's many efficacy and interpretation of results questions that can be raised. What's the optimal duration of neoadjuvant therapy, considering that most of these targeted agents are actually cytostatic by nature? What about variable efficacy of different targeted therapies? We have seen small phase 2 osimertinib neoadjuvant targeted therapy trials that have not met their primary MPR endpoint. So should we just be developing better targeted agents, more powerful agents, or should we get into combination treatment, also taking into consideration that if we do so, then you're potentially introducing sub-studies that are going to be heavier, potentially involving a control arm, so to better untangle what's the contribution of the different combination agents. And lastly, but not least, what's the benefit of the adjuvant portion of therapy as opposed to neoadjuvant alone? Conclusions. Certainly, I think platform trials are here to stay. There's multiple others being developed as we speak, but to our knowledge, CCTG IND242 remains the only cooperative group academic platform. There's certainly a need for molecularly defined platform trials to better define treatment for these patients, and also to better understand how we should treat patients that have both PD-L1 positive disease, but also harbor oncogenic driver mutations, notwithstanding the fact that they may also harbor co-mutation that could be essentially prognostic factors down the line. The final question and suggestion that I would raise is that if you look at the literature, there's been papers published across the way that dealt with general trial methodology. For instance, this one that was published by the MDICT consortium, dealing with how phase 2 trials should be conducted when dealing with targeted therapies. But as can be seen so far, I guess the challenge is that we have the general trial methodology on one hand, but we have also this wealth and exponential knowledge that is disease site specific. So my argument would be to basically better bridge both general trial methodology with disease site-specific knowledge that pertain to early-stage non-small cell lung cancer. So with this, I'm going to stop. I would like to acknowledge everyone that contributed to the IND 242 effort, and most importantly, patients and their families. And I would like to thank you for your attention.
For Pierre-Olivier?
Yes.
Oh, right here. Go ahead, Dr. Milosky.
So what happened to 242A?
Actually, 242A, and I'm allowed to say it now because this was made public recently, the first sub-study had to close down based on a business decision from Novartis.
So why don't you substitute sorafenib? Why don't you substitute another KRAS inhibitor?
That's in development.
Definitely want to do that.
So first of all, congratulations for the wonderful work and the study platforms.
Thank you.
I was wondering if you might want to include also unresectable patients, because we have a lot of problem now for patients that cannot be operated, but doing NGS for every stage 3 patients, we find uncommon mutations, not only EGFR or ALK, and then they need to receive chemoradiation. We know that immunotherapy doesn't work, chemoradiation works. Sometimes we will also need to have a kind of neoadjuvant. So do you think that it would be possible to extend also to unresectable, or in any case to extrapolate the data if the study will be successful for every line, for every mutation? And I do hope so that for every patient you need a drug, and actually you cannot design a phase 3 trial for every mutation. You are right. We will have a problem also to translate all this data to patients that cannot be operated. What is your idea about that?
Yeah, I'm sorry, I'm having a bit of trouble hearing because of the echo, but could you repeat the last portion of your question, please?
No, it's only how do you see the applicability for patients that are not surgical? I mean, because the whole platform is designed around resectable patients, but we have, in any case, a proportion of patients that, for a variety of reasons, cannot be operated, and they have mutations. And what we can do, in your opinion?
If I understand your question correctly, I think that, well, first of all, this is why the clinical trials are needed. In regards to whether or not, if this is what your question pertains to, if we are expecting potential downstaging with targeted therapies that could make those patients potentially operable, I guess it remains to be seen, because so far there's been very little of these neoadjuvant targeted therapy trials being published so far. But I guess time will tell with upcoming published results. And from my take, it probably will be very different depending on which targeted therapies we're talking about, right? We're seeing these incredible path responses with alectinib, for instance, that we're not necessarily seeing with other targeted agents. So yes, I think it's basically with the upcoming trial results that we'll get a better understanding of you know, what is actually needed with these targeted therapies in the neoadjuvant setting.
Pierre-Olivier, if I may, the whole concept of IND 242 was to use the surgical specimen to study clinical correlates of response, resistance, and other biomarkers of interest. So radiation has the unfortunate capability of providing this. So that's what made 242 what it is. If we were going to do the same type of studies to test the concept of radiobiology for one, we would have to think of a different strategy. And it's probably very interesting to look at potential drugs that could be interesting in the radiation sensitizing field. But I think the concept would have to be rethought.
Thank you very much.
Thank you.